guys, this is Hole in Chambers Biology bringing to you Genetics Lecture 2, which deals with Gregory Mendel and his experiment on the pea plants, as well as how we were able to determine dominant and recessive traits, as well as figure out probability of characteristics in future generations. So let's go and talk real quickly about Gregory Mendel. There's a bunch of videos that you guys can actually watch on YouTube that kind of goes over, his history. So he was a Franciscan monk. He's considered the father of genetics because when he was pretty much done preaching, to the community, he would go back and fertilize pea plants, which in turn would produce peas that you could eat. And so he was one of the first to document what these pea plants actually look like as far as traits. And he also was the first to determine which traits are dominant and which traits are recessive. So let's go and talk about why he used pea plants to begin with. So this is actually just a little brief description of a pea plant. They're super simple, and they only have 7 characteristics. And so because the characteristics were very, very simple, tall, short, purple, white, he was able to figure out, generational differences. Now, his control group, now what that means is that he mated 2 purebred tall plants together, and they all produced tall. So no matter what he did, they always produced a tall plant. So we know that that for sure has a dominant trait, okay? He also, in his control group, mated 2 purebred short plants, which we later realized was a recessive trait. But what he knew was we had a tall and we had a short, and they always produced the same short. Okay, so they didn't produce anything else. So then what he did in his experiment is that he decided to take 1 tall and 1 short and mate them together. And so this is considered the pea generation. And again, make sure you guys are stopping the video and writing all of this down in your notebook so that way you can use it during the unit exam at the end of this unit. Okay, so Gregory Mendel verified tall was tall, short was short. So then he mated tall and short together, pea generation, which is known as the parental. And you would think that we'd probably get like medium plants, right? Tall plus short equals medium. Well, that's not really what he got. He ended up getting, as far as his kids are concerned, all tall plants. So he was like, what the heck? Where's the medium? And what happened to the short? Now, a lot of you guys are asking, first of all, how did he do this in the first place? He literally took a paintbrush, took the pollen from one plant, and swiped it into the pollen of another plant. So that's how he was able to for sure know that he mated a tall with a short, because he actually physically did it himself. Well, what happened was he then took the kids. So the kids are known as the Pheno 1 or the F1 generation, and he mated 2 of them together. And so you would think, because the kids were all tall, that their kids would be all tall. But that's not what he found. What he ended up finding was the grandkids ended up having 3 tall and 1 short. So you're like, wait, what? How'd that happen? So the grandkids we refer to in genetics as the F2 generation. So this is a Pheno 2 or grandkids. And this was the ratio. So again, keep in mind that he mated 2 tall kids together. So he mated a tall kid plus a tall kid, and we ended up creating a short plant. So Mendel asked himself, how did the short suddenly pop up after it skipped a generation? So the short actually comes from the original parent, okay, which would have been the grandmother in this case. So then what Mendel did is that he determined that the tall was a dominant trait and short was a recessive. So if you use that as your claim or your hypothesis, and then you write in a tall letter or a capital letter for tall and then a lowercase letter for recessive, all of a sudden we start to realize, well, wait a minute, if the original parents were purebred, that means the original parents way back up over here were big T, and little t, which means that when we mated 2 of these kids together, this child has to only take half from mom, half from dad. So when you look at this, statistically, if this is a purebred, it's purebred homozygous big T, big T. And therefore the mom would be little t, little t. And because the child can only take half from mom and then half from dad, he becomes what's known as heterozygous. So when we mate two heterozygous children together, which you can see down here on the bottom, oops, okay, drop it down, bring it over, we end up creating a ratio, a genetic ratio of a 1 to 2 to 1. So we have 3 that will contain a physical tall trait, and then 1 that will contain a physical short trait. Now again, this is what the expected ratio is. This is not the actual ratio. Okay, so this is what's expected, is a 3 to 1. Now keep in mind, a lot of you guys are going, well, what does that mean, expected, okay, versus actual? So if I, for example, if these were humans, okay, and I made a tall husband with a tall wife, okay, and they have, say, 3 kids. Of the 3, they're expected to get 1 out of 4 chances of a short child, okay? Every time they have a child, 3 out of 4 will always be tall, okay? Now, let's look at some of the laws that Mendel actually discovered by mating these pea plants together, okay? So what did Mendel find? First of all, he found what we call the law of dominance. And yes, this will be on a quiz. Yes, you will need to know this for the exam. And yes, you should probably write this down. Okay, so law of dominance means that some alleles are dominant and others are not. And so, long story short, the capital letter, all you need is 1. If you have 1 capital letter within your genotype, it will show the dominant trait. And that's it. That's the law of dominance. Okay? What Mendel also found or discovered is what we refer to as the law of segregation. And so this is when, from our first lecture, when your parents only give you half of their genetics. That's the law of segregation. Segregation means to separate. Okay? Segregate, separate. So we're going to separate out half your DNA from mom, half your DNA from dad, you put them together, and that creates your zygote, okay? So during reproduction of gametes, what we're talking about here, guys, is meiosis, okay? Parent alleles, which are their DNA, will separate out so that they only put half of their DNA in the egg and the other half in the other egg. So they're not going to give you their full DNA, all right? So again, during meiosis, we create haploid cells, so when the egg and the sperm connect, we will then go back to being a diploid cell. So 23, 23, and eventually to a 46, okay? So Mendel was able to say, yeah, that's true, okay? The next thing that's going to happen, which I'm going to go ahead and present it now, okay? And this is where we're going to be headed. So why is all of this important? Well, eventually we're going to get to a 2 or more trait cross. So instead of just crossing, say, yellow peas with green peas, which is color, you're going to get, say, a green wrinkled pea with a yellow round pea. Okay, and then all of a sudden, instead of having 4 options, 1 and 4, you're going to have 16 different options or possibilities, probabilities of that child having these specific traits every time the parents go to mate. Okay, so please write this down. Law of independent assortment is when 2 or more traits are inherited, alleles will separate independently of each other, which means that they will not affect each other's traits, aka influence each other. So yellow will not influence round. Green will not influence wrinkled. Yellow will only influence the other color, which is green. And the round will only influence the other trait, which is wrinkled, because this is dominant and wrinkle's recessive. Yellow is dominant, green's recessive. So eventually what we're going to see is what we call foiling. Okay, so we're eventually going to look at 2 traits that are dominant recessive. We're going to FOIL out each trait because they're not going to influence each other, so we can now mix them up. Trait 1, trait 2, trait 1, trait 2, and we can start to actually have a variety of gamete options, which in turn will lead us to a large variety of children probabilities. Okay? So please make sure, again, you guys know who Mendel is. Okay, what did he do? He's known as the father of genetics. He studied pea plants. And please make sure you guys know his experiment. So he mated tall with short, came up with all tall, which is your F1. And then the F2 generation is where we ended up getting a 3-to-1 ratio. And make sure also that you guys can prove how that's possible by doing a Punnett square and then writing out your genotype and phenotype. All right, any questions that you guys might have, please make sure to ask me in class. Otherwise, this is Holland Chambers, checking out.